Welcome to STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society. Hi, I'm your host, Dawn Cornegas, and joining me to introduce today's podcast is a man behind the curtain, Dr. Ken Ford, IHMC's director and chairman of the Double Secret Selection Committee that selects all the guests who appear on STEM Talk.
Hi, Don. Great to be here.
So our guest today is Dr. Christoffer Clemmensen, who is an associate professor and lead researcher at the University of Copenhagen's Novo Nordisk Foundation Center for Basic Metabolic Research. Christoffer is a native of Denmark who earned his Ph.D. in Molecular Pharmacology from the University of Copenhagen in 2013. So his lab at the university explores pharmacological and therapeutic treatment for obesity and its related diseases and disorders. And he and his colleagues focus on dissecting neuroendocrine signals involved in coordinating appetite, food-motivated behavior, energy expenditure, glycemic control, and also lipid metabolism. So his lab's goal is to transform molecular and physiological insights into innovative therapeutic strategies that can correct obesity and also its metabolic comorbidities. Joining us for today's interview is our IHMC colleague and senior research scientist, Dr. Marcus Baumann, who is also our guest on episode 116, which we have a link to in the show notes. Marcus is the founder and former director of the University of Alabama Birmingham Center for Exercise Medicine, who joined IHMC last year. But before we get to our interview with Christoffer, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we are especially appreciative of all the wonderful 5-star reviews. As always, the Double Secret Selection Committee has been continually and carefully reviewing iTunes, Google, Stitcher, and other podcast apps for the wittiest and most lavishly praise-filled reviews to read on STEM Talk. If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Today, our winning review was posted by Hamad Adoudi. The review is titled, Awesomely Awesome. It reads, Even Buddha would be enlightened by listening to your guest speakers. STEM Talk is informative and educational. I await with bated breath for each episode. Kudos to you all.
Well, thank you so much for that kind review, and thank you to all of our other STEM Talk listeners who've helped STEM Talk become such a great success.
Okay, and now on to our interview with Dr. Christoffer Clemmensen.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Hi, welcome to STEM Talk. I'm your host, Ken Ford, and joining us today is Christoffer Clemmensen. Welcome, Christoffer.
Thank you. Thank you very much, and thank you for having me on the show.
I'm glad you're here. And also joining us is our special guest co-host, Marcus Baumann.
Hi, Ken, and hello, Christoffer. Happy to be here.
Christoffer, we're really pleased that you could join us today, and we look forward to learning more about your research. But first, we briefly will discuss your background. The listeners always find it interesting to better appreciate how somebody came to be where they are now in their career.
So, Christoffer, I understand you grew up in a small town in rural Denmark in the 80s and 90s. Can you tell us a little bit about your childhood? What was it like?
Yeah, sure. I mean, it was really nothing too spectacular, I would say. It was all in all a pretty great childhood. My parents really had plenty of time for me and my two siblings. They didn't have any big careers, and it seems as if times were a little bit less busy back then, pre-internet and mobile phones. So I think looking back, I feel grateful for having had a childhood back then and as compared to now. So there was a lot of time to play and to do a ton of sports.
And I hear you were an elite tennis player. Can you tell us a little bit about that?
So I think that has to be taken with a grain of salt. So I think it's fair, first of all, to say sub-elite. And second, I think we should also keep in mind that we're a little bit short of 6 million people in Denmark. So tennis is even not a huge national sport here. So with all that in mind, I think I did manage to play fairly well, and I enjoyed to play quite a lot in my childhood and in my youth, and I still actually enjoy the game. So this is something that I still enjoy to this day.
Do you recall when you first felt an interest for science? When did you first become sort of really interested in science?
Yeah, so that came very, very late to me, actually. I think it was actually not until my mid-20s that I really started to become interested in research and science as such. I think if I look back, I can see I did have a personality even as a kid that probably was a little bit kind of permissive, if you can say that. In studying things in much detail, I was obsessing over a ton of topics as a child, but it was not research or anything like that. But I could have long periods of time where I obsessed about like martial arts or skateboarding or something like this. And even, studying it in detail, looking up books at the local library and such. I think somehow personality-wise, I think it was always something a little bit kind of present somehow, that I had a personality for obsessing about details, but it was not until much later in life that I figured out that I could utilize that personality to become a researcher. So it was quite late, I think, as compared to many other scientists.
So as you turned 18, my understanding is you started thinking about going to the university and you thought you would study IT or computer science, but then you went to the Canary Islands just south of Spain, had a change of heart. So what happened?
I think it was a combination of things at the time. I was a little bit lost in terms of, I guess, life direction, and I was fortunate to get a job at this relatively popular sports resort at the island called Lanzarote, and I was there to teach tennis for an extended period of time. And I guess at that same time, it kind of also changed a little bit my life perspective and maybe even self-perception. Somehow this was an international environment, and I guess I grew a little bit of confidence. And at the time, I had also started becoming more and more interested in resistance exercises, or strength training, if you like. And I started also spending more and more time on studying or trying to read up a little bit on exercise biology on the internet, etc. And I think that somehow ultimately altogether gave me the confidence to pursue exercise biology or sports science at the University of Southern Denmark initially. So that was kind of a combination of things that led me to that kind of, as you said, change of heart.
Given all that, how did you decide to attend the University of Copenhagen? Was it sort of a natural option for you, or were there particular factors that drove you in that direction?
So I started at the University of Southern Denmark. So in Denmark, it's a little bit of an interesting educational system we have. So you're admitted to a given university exclusively based on the grades you have in so-called gymnasium, which I guess corresponds somehow to high school. So University of Copenhagen is the most popular university to go to. So people with the highest grades basically it's easy for them to get admitted to that university. And my grades didn't bring me any further than the University of Southern Denmark. But once you first have been admitted to a program, and if you're doing fairly well, you can quite easily transition to another university during the program. So that was kind of, I guess, after a period of time, I think for me, it was a natural transition to change to the University of Copenhagen, also given that they had a little bit more focus on the molecular biology of exercise, which at the time was something I was very interested in. So that was a decision that I felt comfortable with at the time.
That's a great opportunity as well. I mean, they have a world-renowned group in exercise biology there. While you were there, though, you became fascinated by mechanisms of obesity, and that sort of took you in a new direction. How did that change come about?
So I guess in many ways, exercise biology and the science of obesity are not that far apart, right? So there are many overlapping aspects. So at the core, I guess, energy metabolism is somewhat at the core of both of these sciences, if you like. And at the time, I think a lot of the dogmas and or ideas about, for example, calorie restriction, they somehow has also roots or connection to exercise biology, maybe with a little bit of a different focus. But I guess at some point I started to more and more obsess about the many puzzles relating to weight biology, which one way it looks extremely simple, right? So if you consume more energy than you need, that energy must be stored as triglycerides as body fat. And conversely, if you consume less energy than you need, then you must liberate that extra energy from stored energy in the adipose tissue. But that concept is somewhat uninteresting biologically. So the real challenge is, I think, to comprehend how food intake is regulated by the brain and by the body, and the kind of coordinated actions of signals from peripheral organs and the central nervous system in terms of motivation, hunger, etc. So I think somehow the exercise biology and the interest in studying metabolic flux or metabolic turnover led me to reflect more and more about the science of obesity or weight regulation. But there's, I think for me, naturally still linked in many ways, these two fields.
Yeah, I would agree. They're certainly tightly linked. Was there a particular moment or particular event, something that happened that caused you to really focus on weight control, or did this just sort of evolve over time as you were studying exercise biology?
Yeah, I guess they did just evolve over time. It was not a particular moment, but probably a slow transition. And at the same time, I also want to emphasize that I'm still, and my group we're still interested in exercise biology, and we continue to do exercise research in the lab. And we see exercise biology as complementary to studying weight biology, basically. So we're still interested very much in exercise biology.
You received a bachelor's degree in exercise biology and then a master's degree in human biology. What led you to pursue a Ph.D. then in molecular pharmacology? It's a little bit of a shift.
Yeah, so I would like to say that this was because of a big vision or something like this, but the truth is actually it was just coincidental or luck, if you like. So I basically just applied for a series of PhD positions following my master's program, and then ultimately I got one of these positions, and it happened to be in a lab focusing on molecular pharmacology. So fortunately for me, the project that I was going to work on was on trying to uncover an orphan G protein coupled receptor. And I guess for me, the fortunate part was that the receptor seemed to be activated by amino acids, so dietary amino acids. So already here, there was a natural link between nutritional science and or metabolism studying this receptor in vivo. So we had a mouse model for studying the function of this receptor. So it was kind of a little bit of a coincidence that I was entering I would say, hardcore molecular pharmacology lab, but could still kind of pursue physiological regulation of metabolism, given the receptor I was studying was potentially involved in regulation of metabolism in some aspects. So coincidence, luck, and I guess that's it.
Oh, that's interesting. In fact, it seems like a lot of science trajectories go down that path, coincidence and luck. So after completing your postdoc at University of Copenhagen, what took you to Munich, Germany, where you became a postdoctoral fellow at the Helmholtz Diabetes Center?
So there's a small detour to arrive at answering that question. So a part of doing a PhD program in Denmark is that you do an obligatory environmental exchange, and preferably to another country. So that's a part of trying to kind of broaden the horizon of the PhD students in the programs in Denmark, is to expose them to different research environments. So as a student, this is really an excellent opportunity. And at the time, my PhD supervisor was very supportive, and he helped me identify a lab really matching my interest in energy metabolism, and which also went beyond his own direct line of work. So that brought me to Cincinnati, Ohio, where I worked with in the lab of a professor called Randy Seeley for 7 months or so. And here I was joining projects relating to rodent models of this gastric bypass or bariatric surgery, studying some of the molecular underpinnings of the metabolic benefits of the surgeries in animal models. And that shaped, again, my own interest in moving forward or in the direction of obesity biology. And it also created the line of communication with my future mentor in Germany, so Professor Matthias Chöp, who was a good friend of Professor Seeley. So that detour to Cincinnati kind of initiated or created the contact to the lab in Germany, and ultimately that made me then go to Munich to pursue a postdoc. Also within pharmacological treatment of metabolic disease was one of the main focuses of that work.
You eventually became a group leader at Helmholtz, but then transitioned back to the University of Copenhagen. How did that come about? Was that a natural transition for you?
Yeah, so I was lucky to be able to kind of grow in the ranks while I was in Munich, but it was always my dream to kind of come back and establish myself as an independent PI at the University of Copenhagen. And this was fortunately then also made possible by the ability of attracting some of these so-called starting grants, which are research funding that enables you to set up your own lab. So I was lucky enough to get a couple of these grants while I was in Germany, and could use that funding to go back to Copenhagen and set up a lab. So that was a really an opportunity I was all the time hoping to be able to get. And once that opportunity opened up, I just followed through. And so that was great.
So earlier you talked about some of the research questions that you're interested in pursuing. As you well know, good science, especially research that leads to breakthroughs, is driven by people asking big questions. So what would you say is the big question you're pursuing, and what's the question that sort of gets to the heart of your research as you look at your career path?
Yeah, so this is an important topic and something that we think about a lot in my lab. We talk a lot about what questions to pursue and how to formulate those questions and how to also navigate our research constantly toward the most appropriate questions. And I think it's also important to acknowledge that this is not a static thing, right? So every new piece of information we get helps us shape a little bit better the questions that we're pursuing. But all that being said, I think the big question remains as how is body weight regulated? And that is such a big question that in order to try and start to address this, we must, of course, divide it into sub-questions, because in and of itself, it's too complex to just kind of address. It's almost not a question. It's more like an exclamation or something like this. But that's the kind of at the core of our work is basically the regulation of body weight and everything that entails.
That's certainly is indeed a big question. Today, you're an associate professor at the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen, where you head up the Clemmensen Group. On your website, it says that your lab focuses on dissecting the neuroendocrine signals that coordinate appetite regulation, food-motivated behavior, energy expenditure, glycemic control, and lipid metabolism. And that's a lot. Can you give us an overview of this research, the sort of 50,000-foot overview of the research and the lab itself?
Yeah, so I guess on the website, I guess this is also, it is a lot, and I guess it's also probably too broad of a scope for one lab to tackle all of these components. But I think what we've attempted to over the last couple of years is to break down the big question into a few sub-questions. So at the moment, I would say we kind of have three headlines that cover almost everything we do in the lab. And the first one is the endocrine or the hormonal regulation of satiety. And the second one is the role of neuroplasticity or structural or plastic events or changes in the brain in terms of appetite or hunger signals or circuits. And finally, everything we try and do, or every insight, molecular physiological insights we generate, we try and see if these can be exploited for development of pharmacological agents or putative new treatments for predominantly obesity. So these are, again, I guess, three big topics, but these are the topics that most of our projects revolve around. So as a research lab, like most labs, we fluctuate a little bit in lab size with students coming in and out. And at the moment, I think we're probably around 15 people in my lab, including students. And each person has his or her own project, and they all then typically relate to one or several of these before mentioned themes that we work on.
So obesity and its related diseases like type 2 diabetes, cardiovascular disease, as we all know, these are serious problems affecting worldwide public health and also the global economy. You pointed out in a 2019 paper, which was titled Emerging Hormonal-Based Combination Pharmacotherapies for the Treatment of Metabolic Diseases, that the treatments we've been using to deal with this problem have not been able to really effectively reverse the staggering rates of obesity. Can you talk about how this paper underscored the need for more effective treatment approaches?
Yeah, so at the moment, there's quite a lot of progress in the treatment of metabolic disease, and slowly there's becoming regulatory approval of more and more efficacious and safe drugs. But despite that we're witnessing this progress, we're nowhere near being able to kind of halt the global burdens, these dual burdens of obesity and diabetes and the associated complications. And this is much more complex than just observing or kind of noting down results from controlled trials. So one thing is to develop new and effective and seemingly safe therapies, but there's still a long way to go in terms of this having an impact on these huge burdens on societies worldwide, as well as, of course, obviously economies. So that was at least one of the kind of background rationales for diving into this paper and looking into the future of treatments for the metabolic diseases.
In that paper, you write about the need to target multiple signaling pathways if we ever really can hope to see significant improvements in this whole body weight issue. Can you give us an overview of the hormonal-based pharmacotherapies that you've been looking into as potential treatments? I think the listeners will find that really interesting.
Yes, so the idea is basically that body weight is very, very tightly controlled, or even you can say protected by the organism. So most people will realize, even though they don't count calories, that their weight is remarkably stable over time. And this also implies then that any given pharmacological treatment trying to, if you like, disturb this balance must be potent enough to overcome these biological mechanisms that ensures weight stability. So we talk about counter-regulatory biological mechanisms. So any insult initiated to try and, for example, create a negative energy balance to reduce body weight, that will immediately try and be accounted for by so-called counter-regulatory biological mechanisms. So let's say that you target appetite. We oftentimes see a corresponding decrease in energy expenditure, which is one way the body will try to kind of mitigate this insult. So one strategy would be to try and devise therapies in which non-overlapping pathways are targeted in parallel. So for example, both targeting appetite and at the same time targeting energy expenditure. And most of these strategies that are looking into this type of combinatorial targeting are based on gut hormones, so factors that we have identified being secreted from the gastrointestinal system, typically in response to a meal. And these factors has then over the last decades been optimized through medicinal chemistry approaches, for example, giving them a better, like a pharmacokinetic profile. So they have an increased bioavailability and can thereby be used as drugs. So that is the objective by these hormone-based combination pharmacotherapies.
You may have actually touched on this partially just now, but I want to talk a little bit about a really important problem, which is that the overwhelming majority of people who do manage to lose weight end up gaining it back. So why is it that people struggle with this, and what are the most effective therapies to help people keep the weight off?
Yeah, so it goes hand in hand with what we just talked about. So basically, we must come to the acceptance that all conscious or voluntary attempts to modulate our body weight, for example, by going on a diet or exercising, that is easily corrected by these homeostatic forces or these counter-regulatory mechanisms. It's the autonomic processes that are basically governed by our central nervous system and predetermined to a degree that ensures an individual target body weight. And this originates from traditional gene by environment interactions. So no matter how effective any given diet is, for example, for a long time, we've seen a growing interest in low carb and or ketogenic diets and their success in, for some people, creating a negative energy balance. But ultimately, they must all face the same counter-regulatory biology. So the biological counter-regulatory mechanisms, they don't really care about what diet is responsible for a negative energy balance. They'll just try and fight that with all the power that is inherent or that is available to our biological system. It seems then as if few individuals are fortunate enough to be kind of, if you like, predisposed to be able to follow through with a permanent lifestyle intervention, and likely because they maybe hit a little bit less hard by these counter-regulatory mechanisms.
The counter-regulatory mechanisms that you're talking about, I think, are powerful forces, as you said, and this whole issue of satiety and what is satiety to one person versus another. I think that plays a big role, and it's a difficult thing to tackle. So you followed up your 2019 paper with a review in PLOS Biology that looked at the evolutionary and environmental perspectives on body weight regulation. So we love one of the questions you asked in this paper, which was, why are we not all obese? It's a great point. So let's sort of ask another question. Why aren't more of us obese? And why aren't we all sort of prone to gaining weight? There certainly are people less prone than others. Do you have any comments about that?
Yeah, so that's a super fascinating question. One of the things that we talk about the most in the research we do and in the lab in general. So, I mean, it's a big mystery that even in the most quote-unquote obesogenic environments, that there is a fraction of the population that remain remarkably slim. So even some individuals keep the BMI below normal weight without having any type of eating disorders. So some people have a biological system that protects them from gaining any weight, and despite whatever environment they're exposed to. So part of the answer to this variability in our body weight likely lies in our individual genetic predisposition. So typically based on twin and family studies, estimates suggest that up to 70% of our variability in body weight can be explained by genetic factors. So some individuals will have a more vigorous defense system against weight gain than others, whereas it seems as if the mechanisms that protects us, if you like, from losing weight, if we go on a diet, for example, we're all equipped with a very efficacious system that unfortunately defends us from successful dieting. But on the other side of the coin, the mechanisms that protects us from gaining weight, there seems to be much more variability from individual to individual. And that likely explains the big variability in body weight we see today. And it's been suggested that since our ancestors climbed to the top of the ladder of the animal kingdom, that was the time where they also escaped predation. There was no longer an evolutionary pressure for keeping weight low, so there was no predation risk. And since then, it could be that random genetic mutations have contributed to this picture we see today with this big variability in this, we call it the upper boundary of kind of weight balance, or the upper intervention point where the body starts to fight against the weight gain. So it's clear from overfeeding studies that we are equipped with a system that protects us from gaining too much weight, but this system seems to be very different between individuals. So this model is sometimes referred to as the dual intervention point model, and this is a theory that's really championed by a guy called John Speakman.
One thing, Christoffer, is it to me seems unlikely that we experienced as a species a worldwide evolutionary change in the last 30 years. So if you just look back 30 years ago, or look at photographs of people in World War II, which is really recent, they're all very thin. And so I have doubts about a recent biological change.
100% agree. So I think it's important to emphasize that our genetic predisposition to develop or to develop obesity has always been there, but it requires an environment that is permissive to unleash those susceptible genes. So once you put people with genetic susceptibility to develop obesity in a modern obesogenic environment, you'll see that up to 2/3 of the population will develop a BMI up to 30 or above. So it seems if the genetic predisposition is likely to have been there for hundreds and hundreds of years, but it's only recently that we've engineered a society that is really, really bad for us in terms of kind of unleashing those genes.
Yes, for sure.
So what about the elusive slimming gene? I think this is, based on what you just talked about, a highly relevant topic. So researchers have identified, a number of agents that keep our fat mass from becoming too low, but have had trouble understanding the mechanisms by which we regulate excess fat. But there is some evidence dating back to the 50s, sort of for the existence of potentially blood-borne weight gain preventative molecule or multiple molecules. Can you talk about that?
Yes, so it again relates to the fact that we see as part of the population that managed to remain extremely thin in obesogenic environments. And the fact that once we introduce both animals and humans for these controlled, we call them overfeeding trials or experimental overfeeding, we do see that, yes, we can make everyone gain weight in agreement with the laws of physics, right? So everyone will gain weight if we give them more energy than what is required for them to remain weight stable. But at the moment we then release people again to their free living ad libitum conditions, we see that they quickly, bounce back to the pre-intervention body weight, so they lose weight again. There's a little bit of a conundrum in context of the modern obesity epidemic, but this is what we see under these controlled experimental settings. And this is what kind of beautifully illustrates this biological defense system against weight gain also. But it seems as if some people are much more tightly regulated in terms of not gaining weight, even under these free living conditions. So it seems as if this boundary that is preventing people from gaining weight and forcing them back down again is much lower for a few or a subset of individuals. And I think now at the moment it's being debated whether, or it's likely that there is also genetic predisposition that can be identified for these slim individuals. And that is really interesting area of research to try and identify their genetic predisposition as well as their physiological mechanisms that protect or defend against weight gain and keep them very slow in a modern environment.
We've talked about several of them, but are there other factors that you think are important in regulating energy balance in the body? What are some of the, you mentioned hormonal, we've mentioned genetic. What do you see as the big 3 or 4?
Yeah, so that's a big topic. I mean, I think it's clear that we must acknowledge that the brain is primary regulator of our body weight. So also if we look at the current genetic evidence of body weight variability, all of the genetic mutations that are associated with differences in BMI or weight are all related or encoding for factors in the central nervous system or in the brain. So it's clear from many angles that the brain is really the primary organ to look at in terms of weight variability. But of course, then the brain is not a kind of a standalone organ, right? So it constantly receives information from what we typically refer to as the periphery. So that is both hormonal cues, circulating nutrients, as well as direct neuronal signals. So there's a constant cross-communication between the brain and the remaining organs of the body. Those are the signals that we try and decipher and kind of rank order in terms of their importance. But I guess none of them, it's difficult to single out some factors. Of course, some are more potent than others, but the complexity is pretty vast, right? So we haven't even touched upon the microbiome, but that in and of itself is a huge research or focus area at the moment that also is being looked into as a part of this whole body integrated system that regulates our weight.
So maybe we should be looking for mutations or epigenetic modifications in these sort of hot dog eating champions, these people that just consume enormous amounts of calories and remain relatively lean. So
it might have to do with what happens after they eat the hot dogs.
Yeah, I would imagine
they might
so.
Not stay with them.
Right.
At the end of the day, a chronic state of positive energy balance certainly leads to weight gain. So in your view, what is the key pathway here to target? Should we target appetite control or energy expenditure? You see people camped out on the elliptical machine for hours and they look the same always. Or is it some combination of both? What is your take on that? I know it's more complicated than this, but just what is your take on that question?
Yeah, so the quick and straightforward answer is both. But I would like also to, I guess, rank the answer in saying we must target appetite control. It seems as if all therapies targeting exclusively energy expenditure will be quite quickly accounted for by an increase in appetite too, again, as a part of the defense system by the body. So targeting energy expenditure is unlikely as an exclusive indication to create a chronic negative energy balance. So it seems as if appetite lowering therapies can in fact create a substantial weight loss. And we see some of the current therapies, some actually just one therapy just being approved by the FDA a couple of weeks ago is exclusively targeting or inhibiting appetite. And that can actually create weight loss between 15 and 20% in subjects with obesity. And that's not an irrelevant weight loss, I would say. And that's by exclusively targeting appetite. But if you think about adding then a small or slight increase in energy expenditure, maybe we can actually reach, 25% or so weight loss. And that would actually be remarkable.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research lab pioneering groundbreaking technologies aimed at leveraging and extending human cognition, perception, locomotion, and resilience.
We explained to the listeners the remarkable differences between brown and white fat, and in this context summarize your interesting 2018 paper in Nature Communications describing the potent effects of stimulating brown fat thermogenesis with isilin.
Yeah, so that's another really important and I think also kind of popular research topic at the moment, and that is the ability to target subset of fat cells to increase their energy expenditure or to ramp up energy turnover in these cells as a part of a mechanism to ultimately create a negative energy balance. And there are these subset of fat cells called the brown fat cells that have a higher thermogenic capacity, and their main function is likely to, as a thermogenic organ, to elicit heat production, as opposed to the white adipose tissue, which traditionally has been viewed as a more energy storing organ. But it's not as simple as that nowadays. I think it's also important to acknowledge that the white adipose tissue is probably the tissue responsible for secreting some of the most potent appetite regulating hormones we have. But the interest in targeting the brown adipose tissue is definitely the idea of ramping up thermogenesis, if you like, to create a negative energy balance. And identifying compounds that either directly or indirectly will increase brown adipose tissue thermogenesis is really something that is being worked on quite a bit at the moment. There's one caveat, and that is still being debated, and that is the amount of relevant thermogenic adipose tissue humans have as compared to some of the model systems that we often study, which are rodents that have substantial amount of brown adipose tissue.
We've also here at IHMC have been interested in brown adipose tissue and how you might enhance the thermogenesis function, not with respect to weight loss, but with respect to tolerance of cold water or cold immersion. We find it quite interesting as well.
Yeah, and I think that's also just to add to the isilin component that was mentioned before. So isilin is a synthetic ligand that's been developed to target a cold sensing receptor, a cold sensing ion channel called TRPM8 or TRPM8. So that's basically a part of the natural temperature sensing system of the body. Is we have these cold sensing receptors that signals to the central nervous system to engage a whole cascade of adaptive mechanisms to basically, again, fight against this homeostatic temperature challenge. So by developing ligands that more effectively directly target this type of temperature sensing channels, there's also the TRPV1, which is a capsaicin or chili receptor, which is a heat sensing receptor, is also interesting areas of research, also in terms of therapeutic utility.
Very, very interesting. So geneticists, as you know, are pursuing and starting to uncover sort of weight gain defense genes. In your PLOS Biology paper recently, you argued and sort of talked about the fact that for humans to fully benefit from such genetic research, physiologists will have to play a role in the interpretation of these discoveries. What kind of collaboration between physiologists and geneticists do you anticipate becoming necessary as we go forward?
Yeah, so this is really an extremely important point, and that's something that we have more recently been actively pursuing to engage in these type of cross-disciplinary, if you like, collaborations to an extended degree. So fortunately, at the center where I'm at now, we have a huge tradition for looking into the genetic components of metabolic disease. So we have world-class researchers, genetic researchers, and we have an increasing amount of collaborations between the physiologists, geneticists, molecular biologists, and also even epidemiologists. So let's say if we want to identify a subset of extremely thin individuals, we want to be able to recall these individuals into our facility. We want to be able to genotype them and then subsequently also phenotype them. So by physiologically, molecular biologically. So that is one way of really using or combining genetics with physiology and even epidemiology and etc. So I think that is ultimately the approach that is needed to reach to the next level of understanding of, for example, weight biology, and then ultimately combining this with some of these more modern, oftentimes referred to as omics methods that will allow us to, in a more unbiased fashion, to detect thousands of, for example, circulating factors.
Mm-hmm. As we discussed earlier, the human genome has largely been stable throughout this really remarkable rise in obesity over the last several decades. We touched on this earlier, but I want to loop back to it. I mean, this seems to sort of suggest an epigenetic modifications may be playing some kind of an important role here. And so I wonder, to what extent do you think differences in obesity susceptibility that you talked about earlier, that we see across individuals, might be explained by differences in the epigenome as much as in the genome?
That's a good question, and I'm not an expert in epigenetics, but I will say that this is something that we are also interested in, given our interest broadly to understand the biology of weight regulation. So I think one angle that we haven't talked about or touched upon is the contribution of socioeconomic factors to obesity or weight biology. So there is a clear relationship between socioeconomic status and obesity levels. So how do we reconcile this with what we know about genetics and environment? And one way of approaching understanding this is we could talk about mechanisms of stress or psychological challenges and how that is feeding into some of the more well-known mechanisms of energy homeostatic control. Or it may also again relate to epigenetic mechanisms. So there are historic examples of how, for example, there's this example of this so-called Dutch hunger winter, where it's been identified in subsequent generations that they had altered sensitivity to develop, for example, metabolic disease and other diseases, and that kind of corresponded to the time of conception in terms of global stress on a given population. But that's, of course, again, we're linking. These are kind of observational, epidemiological observations, and then trying to take this knowledge and to conclude something causal is of course also a well-known and classical challenge we have in research. But it's definitely interesting to try and take all these mechanisms we've talked about into account to try and make one big coherent theory of weight regulation.
So we talked earlier about a number of these issues, and it seems like you put a lot of stock in and feel strongly about regulating appetite control. So what is the sort of current status of pharmacological obesity treatment, assuming appetite control is a major player in that? How do you see the landscape changing as we go forward?
Yeah, very timely question, given that I think I just highlighted, 2 weeks ago, the FDA approved a new drug for the treatment of obesity called semaglutide, which is a glucagon-like peptide 1 receptor analog. And in short, it's a compound that mimics a hormone in the gut called, in short, GLP-1, the abbreviation. And that hormone is typically reaching two areas of the brain involved in appetite, namely a region called hypothalamus and then a couple of regions in the brainstem. And this system is very well described biologically, and through years or decades of optimization, a couple of companies have really managed to take this molecule and modulate it in a way that it's become really a super attractive or potent agent in terms of appetite suppression and therefore also weight loss. So we now see the recently approved drug, we see that a weight loss between 15 and 20%. And this is developed by actually a Danish company called Novo Nordisk, but we also see Eli Lilly coming with another type of molecule that integrates this new generation of molecule, these dual agonists. So in addition to the GLP-1, it also brings another gut peptide called GIP, and they've kind of created a promiscuous molecule that targets both of these receptor systems. So we both see in this drug, the rationale is that you'll see enhanced glycemic or control, that means added benefits for type 2 diabetes, and you retain this potent weight-lowering effect from the GLP-1 component. So this kind of is a next generation of anti-obesity drugs where we see weight loss beyond 15%, which is a little bit of a game changer, because up until now we've typically seen maximum up to 10% weight loss, and coupled with a series of adverse events, which seems less with these type of new therapies.
Discoveries, we hear about them all the time in the headlines. particularly in discoveries in mice. And the media picks it up, and they highlight these flashy headlines, typically without mentioning mice. But as you know, mitigating obesity in mice or cancer in mice or anything else in mice is many steps away from demonstrating efficacy in humans. What are the current challenges for the translation of promising results that are discovered in rodents to humans in your field, in the field of obesity research? Are there particular challenges there, or is it just the normal challenge of translation from animal to man?
Yeah, so that's good point to discuss also. So I oftentimes say that I've cured mouse obesity repeatedly, and I can easily create a new therapy that will cure mouse obesity. But no, that being said, in general, the rodent models for metabolic disease are quite good if you compare to other types of areas of research. So in that way, we are kind of fortunate or quite lucky. That being said, there are some aspects of the rodents that actually makes it a little bit complicated. So one, it sounds a little bit bizarre, but the fact that rodents actually they're not capable of throwing up or vomiting, and one of the primary adverse events of most weight loss therapies is actually nausea and vomiting. So that is one component, or that's one adverse effect that is difficult to pick up in the rodents. So it's tough to see when a given efficacy is being outmatched by an adverse event that would be not tolerated in a human situation. Adding to that, there is the energy expenditure component. We talked a little bit about it with the brown adipose tissue. So the rodents have substantial amount of brown adipose tissue, and targeting thermogenic pathways in the rodents is a very potent way of creating a negative energy balance. In humans, in my opinion, that has yet to be demonstrated that is an equally relevant target. So those are some of the aspects that are slightly challenging in terms of translation. But in general, we are somewhat fortunate, I think, working in the metabolic disease field, that the rodent models are quite representative for what we see in humans.
One of the things that I think causes some confusion, not with someone like yourself, of course, but in the mainstream media, is you'll read about a high-fat rodent diet, and then you see, yes, it's high fat, but it's also high sugar, and not at all like a high-fat diet in humans. And I think this causes media confusion in particular.
Yeah, so that's another thing, and we haven't touched upon that component, and that is studying specific macronutrient distribution in rodents and even the addition of specific micronutrients. I think that's also an area of work where there's a risk of losing a little bit of information in translation here. So yes, so oftentimes when we try and develop new therapies, weight loss therapies, we exclusively use this combined high-fat, high-sugar diet in the rodents to get them basically very fat and to make them metabolically compromised. So for us, it's a strategy to make a window for which we can look at a weight loss. But I do agree that we oftentimes refer to the rodent high-fat diet too simplistic, as it oftentimes also bring a lot of carbohydrates in the form of refined sugar, etc.
Mice love donuts too.
Yes, they do.
While we're on the topic of things that can be confusing, one of the things that always jumps out at me is what seems to me like an over-reliance on weight and BMI as opposed to body composition. And you know, so you see a tremendous focus on weight loss as opposed to composition. And I think muscle is a very active endocrine organ and a store of glycogen. And so I think that gets underappreciated, particularly in the sort of mainstream headline media.
Again, 100% agree. And now we started the conversation talking about exercise biology. And I think fortunately with the background in exercise biology, we are a little bit more aware of body composition as a key component to look at when examining changes in weight or BMI, given that a lean mass is desirable in large amounts as a contrast to body fat. Again, this is an area that is also being looked at quite extensively also in terms of the genetic aspects. So genetic predisposition for changes in body composition is also an interesting area of research. And again, I agree, in the rodent models, there's something that we often don't pick up on because weight loss studies in rodents are typically over a few weeks. And here you induce a dramatic weight loss and there's no the sensitivity to pick up changes in body composition is typically not there. So that require more long-term human studies to really scrutinize differences in body composition. And yeah, talking about just BMI exclusively can definitely be misleading.
This leads very nicely into the next topic. Sort of going back to the FDA, the newly approved FDA drug for GLP-1 biology, and thinking about appetite control, 15+% weight loss certainly is a game changer. But what do we know about long term? So if an agent such as that is used, is it going to be successful over the long term as a pharmacotherapy? And I'm talking, years rather than weeks or months. If that pharmacotherapy approach is in the absence of any real attempts for behavioral change?
Yeah, so the short answer is, at least to my knowledge, we don't know. So I haven't seen any studies with these new treatments that go beyond 1.5 years. But what is clear already from the published studies is the moment you take away the drug, individuals will immediately rebound their body weight. So once you remove the drug, the weight will go up again immediately. So it corresponds to any other lifelong treatment, such as cholesterol-lowering drugs or blood pressure-lowering drugs. It's a drug that you need to take lifelong to continue to work against your biological feedback or homeostatic system. So I guess the holy grail would be to create a therapy in which the defended level of body weight, or we talk about this set point mechanism, is lowered, so you defend a lower level of body weight instead of working against a weight. So that is a distinction that is, at this point in time, it seems a little bit far away, but there's something that I think we are working toward, is to continue to try and explore ideas that would lead to the development of a drug that will lower the defended level of body weight. And this is what we see following the bariatric surgeries, so the obesity surgeries. We see that following this surgical intervention, that the individuals actually defend body weight that is typically 30 to 50 kilogram lower than what they had pre-surgically. So it feels effortless to keep that lower body weight, as opposed to going on a diet or as opposed to the treatments that we have available right now.
So in terms of future research, you and others stress the importance of omics as a path forward, proteomics, genomics, transcriptomics, metabolomics, etc. Could you elaborate on how this is revolutionizing the field, as well as how you are currently using omics approaches in your own program?
Yes, so again, I don't know if I'm the right person to talk exclusively about omics, but we are using it as a tool, and we try to use it to learn something new. But I think it has yet to show that they can actually teach us something revolutionary new about biology. So we see an increasing amount of studies in which these omics methods, metabolomics, proteomics, peptidomics, etc., are being used in an unbiased fashion to try and learn something new about a given tissue or circulating factors. And we see thousands and thousands of signals coming out, and we can rank order them. What we oftentimes find, at least in our hands, is that the factors we already know are typically the top hits in these unbiased screens. And that's, of course, reassuring, because it's kind of a positive control. You find what you're expecting to find. To my knowledge, these methods have not led to the discovery of a new important factor yet, but that is, of course, what we're hoping for. So let's say we try and scrutinize in an unbiased fashion the circulating proteome, so all the proteins in circulation in response to a given intervention. The idea is, of course, if you can link a circulating protein that could be a hormonal-like signal with a given intervention, that you could identify a new factor that is important for example, in our case, it would be weight biology. So that is the hope or the dream, but I think we must still be skeptic in terms of our ability to use the big data and to interpret the data. And also, I guess the sensitivity for some of the methods are still not quite high enough to reveal all the factors that we wish for it to reveal.
Our earlier discussion of blood-borne weight gain-related molecules brings us to an article that you published later on in 2020 addressing molecular changes in the body following bouts of endurance and resistance exercise. A particularly interesting aspect of the study is the untargeted nature of the metabolomics profiling that you just mentioned. Can you explain the greatest benefits of an untargeted approach. So you started on that just now. The untargeted approach has the benefit of sort of lack of bias from one perspective. But you also mentioned some major challenges. And I don't want to dig deep in this, but just briefly, if you could talk about what you see as the primary challenges. You touched on a couple, as well as the benefit.
Yeah, so I think one of the challenges that the field is facing is the urge or the dream to single out factors or a few factors exclusively and to then utilize those for further therapeutic pursuit. So there is this dream of exercise mimetics that, you study exercise to a degree that you can develop a pharmacological agent that will mimic exercise. So you don't have to exercise, you can just give a drug. And I think looking at the available data, we just exclusively by looking at the metabolome, we see 800 metabolites being affected by exercise. So I think it's a little bit naive to think that we can create a therapy that will mimic exercise, giving countless of molecular and physiological adaptive responses we see in response to just a single exercise bout. Of course, the reason why we're still doing this is, of course, I think these analysis can give rise to new ideas, or they can be the starting point for new explorative therapeutic pursuits. But there's, I think, sometimes a little bit of a mismatch between wanting to use these type of studies to single out factors that can then mimic some of the utility or some of the benefits of exercise. So, for example, in our unbiased approach, we see some of the top hits are, for example, lactate or succinate or alpha-ketoglutarate. And some of these factors they're all very well known already. So it's not as if we reveal new highly regulated circulating metabolites following exercise. So we see the usual suspects, and we of course see them in context with a lot of other interesting signals. But transforming or taking this knowledge and try and, for example, start injecting or administering these single metabolites in hope that this will create some of the benefits of exercise, I think is probably not gonna materialize into something that's relevant therapeutically.
Since there are so many metabolic changes measured and identified, how did you decide which metabolites to study more closely?
Yeah, so that's actually, we almost gave up on doing this because, as I just mentioned, it seems a little bit naive to single out factors and study them exclusively. So for the exercise biology, I think this is actually not an angle that we will pursue further. I think we're happy about the descriptive work and kind of presenting the entire picture. But when it comes to weight biology, I think we still hope that we'll be able to identify factors that could be a little bit more relevant or potent as a standalone factors. But in terms of exercise biology, I think at least our lab, we're not going to pursue, based on this work, single factors to follow up on, given that actually the factors that we saw most abundantly regulated are already well-known factors.
That makes a lot of sense.
Yeah, and I fully agree with you. I think exercise is such a multipotent, you could almost call it a pluripotent stimulus, that mimicking it with one or two targets is just not realistic. But in this interesting study, you focused on the acute molecular and physiologic changes that took place, within a short window post-exercise. And as you know, there are efforts underway to better understand these acute changes in the context of longer-term training as well. The acute responses to exercise in a sedentary human or lower animal, frankly, are likely much different than acute responses in trained individuals. So any thoughts on how we can learn from the range of responses in untrained versus trained people as it pertains to weight management mechanisms or, other health benefits as well?
Yeah, so I think that would actually be interesting. And that's something that we also highlighted in the manuscript, that this is just one set of individuals, and actually the individuals we studied here were actually quite well-trained individuals that had experience both with endurance exercise as well as with resistance exercises. So they were adapted to both types of exercise. So we didn't look at the immediate stress response to a new mode of exercise. But I would be very excited to see similar setup in untrained individuals as well as individuals that are metabolically compromised, for example, with the high BMI or with different stages of type 2 diabetes, or we haven't even also started exploring sex differences, etc. So I think those type of comparisons, again, they're descriptive, but I think actually that type of descriptive work is relevant to better understand how exercise affects different individuals differently. And I do believe that comparing these responses would actually provide some new information. But this is, to my knowledge, this has not been done. And I guess it would make sense to do it all at once for the bioinformatically, so everything could be compared at the same time. But this is also there would be a big research effort.
This brings us to your most recent paper that was published earlier this year in Nature Communications, where you asked the question, how does the exercise and appetite-regulated protein GDF-15 act in different contexts? You found that it acts differently based on whether it's endogenously produced or pharmacologically applied. Prior to publishing this paper, it seems that very little was known about the effects of endogenous GDF-15. Can you talk about that?
Yeah, so GDF-15 is a very hot topic in metabolism right now. So the reason why I guess that is that in 2017, several big pharmaceutical companies published more or less simultaneously research papers identifying the endogenous or the receptor for GDF-15. So the so-called de-orphanized receptor, it's a receptor called GFROL. They showed that this is expressed in the brainstem, and once you create, through means of medicinal chemistry, GDF-15 or GDF-15 analogs and activate the GFROL receptor, you see a weight loss in animals such as rodents and non-human primates. So GDF-15 was discovered already in 1997 and initially kind of considered as a type of inflammatory response. And for many years, it was kind of a little bit flying below the radar, at least of researchers in the metabolism field. But those studies in 2017 really put it on the map as a relevant candidate for modulating energy balance. So that escalated and kind of created a storm of new papers on GDF-15. We also, we had some ideas which ultimately made us also kind of jump the GDF-15 train and started studying it. So our way into it was a little bit different because we actually noticed that GDF-15 was increased in response to exercise. So as you just mentioned, endogenously, it's being produced once you subject people to endurance exercise. So that made us speculate about why that is, and try to understand it a little bit better. We followed up more recently where we did a series of different types of exercise. We had some samples that were available for more long-term exercise bouts, including marathon run and long-term cycling, etc. And we see actually the hormone goes up many fold following these type of long-term interventions. And given that we know that the pharmacological administered version lowers appetite, for us that was an interesting angle to try and understand further and to test the idea that could the exercise-induced GDF-15 actually be a part of this acute suppression of food intake, which is actually a phenomenon that is quite well acknowledged following vigorous exercise. You see this acute suppression in appetite, and you even see reports of nausea, etc. Could GDF-15 be the signal mediating that? Was what we set out to understand in this paper. Long story short, we didn't find evidence for that, and we think the mismatch between the endogenous GDF-15 and the exogenous or the pharmacological GDF-15 is to be found in the levels that we observed. So when you give GDF-15 as a pharmacological agent, we see many 100-fold increase in levels as compared to what we can ever create endogenously. So that's kind of, I think, dampened our interest a little bit. But again, this field is moving kind of with a fast pace. So there are now indications of GDF-15 receptor, the receptor called GFROL, but there are indications in the literature of GFROL independent effects of GDF-15. So it might be locally produced GDF-15 could have what we call paracrine or autocrine effects in the venicity of where it's being produced. So I think we remain agnostic to whether the endogenous produced GDF-15 can have an impact on energy metabolism, and we continue to work on this with great effort and enthusiasm.
Mm-hmm. Sounds like your body's way of telling you to stop doing that.
So that's exactly, at least that's how the signal seems to be produced in terms of a broad amount of diseases. So many diseases, you actually see GDF-15 as a clear biomarker. It typically increases in plasma in response to all kinds of organ or cellular stress. And this could also be the circumstance in exercise. It's kind of a feedback signal or feed-forward signal, if you like, to the central nervous system to tell something is wrong, stop what you're doing, as a kind of a generic stop signal. And I think that's currently what's agreed upon in the field. But there's still a lot of details to understand. There's some mismatch, in our opinion, pertaining to pharmacological versus physiological GDF-15.
So there are a number of exercise response molecules, whether they be hormones, growth factors, metabolites, so forth, that are intensity dependent or dose dependent, right? So modes of exercise versus intensity within a mode of exercise. Does GDF-15 have any dose dependency on how much it rises post-exercise, both in magnitude as well as time horizon, how long it remains up?
Good point. So studies we've done, it seems as if you must exercise beyond 2 hours. It's not a magical time point beyond 2 hours, but sometime around 2 hours of relatively high intensity endurance exercise, such as up to, let's say, 70% of your VO2 max or similar, is required to reach a level where you see a steep increase in the endogenous production. But if we look at subjects that has been exercising for just 45 minutes or so, we see a robust statistical increase, but its magnitude is really subtle. But if we look beyond 2 hours, we see this many-fold increases. There seems not to be anything related to training status, whether you're untrained or trained. And intensity, I think we don't know at this point, but my guess is that you need to be exercising at a relatively high intensity. But again, this is really, there's not many groups working on the exercise biology of GDF-15. So we'll see future results coming in this area, I'm sure.
Yeah, I think, this whole concept of intensity thresholds for various signals, we have a lot there to learn. And I think this is just one example, but I think it's an exciting future for the field to really hone in on what intensity is required to get what biologic effect, which sort of raises the question about the public health guidelines to just go out and walk your dog more versus, higher intensity training that might be required to induce some of these pathways. But
And that's one I don't want to induce. The GDF-15, I'd be happy if I don't do that.
But I think there are probably a number of signals that are intensity dependent in a powerful way that we overlook sometimes in public health messaging to just walk more.
Yeah, or it's seen as a virtue to camp out for an hour and a half on the elliptical machine while watching television.
Fair enough. Christoffer, this has been a fascinating discussion. Your research is definitely gaining attention. Your lab has grown. You must feel good about the growth of your program. And, we'd love to hear about where it's headed. What are the new funded projects that you have?
Thanks. And I think we're quite pleased with where we're at now as a research group. So I started the lab in Copenhagen in 2017, and now it seems as if 4 years or so later, a lot of our projects are really entering a stage now where we see a lot of exciting results emerging. So this is a super exciting time to be scientists. So I'm pleased, and hopefully lab members are equally pleased. Yeah. So I think we talked upon a little bit the directions that we headed already in the lab, and I think we just continue toward studying some of these big questions. I mean, identifying new regulatory pathways of satiety is a big question for us. We didn't talk too much about these plastics and changes in communication between synapses in the brain in terms of appetite control. This is something that we're very excited about. And finally, we have increasingly delving into medicinal chemistry. So we make these Trojan horse-like molecules where we conjugate peptides with small molecules to specifically deliver these small molecules to target more potently and specifically different pathways that we're interested in. So a lot of exciting things going on at the moment. So yeah.
This has been a really great science discussion. I'm sure the listeners who have learned a lot as we have. Switching gears a little bit, I understand you and your wife have 2 young children. I would imagine they keep you both very active and busy. Other than keeping up with the children, what else do you do to stay fit, stay in shape? Do you still play tennis?
I do. I play now. I'm at an age where I play in a kind of I guess it's called veteran tennis or old boys tennis. So I enjoy that now and then. But I guess in addition to that, I try now and then if there's a 15-30 minute gap to do either a quick run or a little bit of resistance exercise. I actually subscribe to the idea that just doing a few pull-ups or a couple of sets of deadlifts or bench press is substantially better than doing nothing, obviously. So I try and do this occasionally.
Before we let you go, I understand that this may be your first podcast and interview in the English language. And by the way, you did a great job. Is that right? And do you listen to podcasts or no?
This is, in fact, the first English podcast interview for me. I've been doing quite a few in Danish, and I'm now I'm happy that you invited me onto the show, and that this was my first one in English. I absolutely love podcasts. I listen to them on the go all the time, both science-related podcasts, but also more entertainment type of podcasts. For me, it's a super nice way of combining transportation with something that both can be educational and or entertaining.
Yes, I feel the same. And so, Christoffer, thank you so much for joining us here today on STEM Talk. You're a lovely guest. We appreciate it.
Thank you.
Absolutely. Really enjoyed it. And thanks again for taking the time to share your research with us.
My pleasure.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. STEM Talk.
So Ken, I'm definitely sorry that I missed this interview, but Marcus was definitely the perfect person to co-host today's conversation since much of Christoffer's research focuses on changes that take place in people as a result of exercise. And for those listeners who may have missed our interview with Marcus in episode 116, Marcus is internationally known for his research into the biological mechanisms underlying exercise-induced adaptations, and also their clinical utility in disease prevention, treatment, and also rehabilitation.
You're right, Dawn. Marcus was indeed a good fit for today's interview. And Christoffer is a young, up-and-coming researcher whose lab is conducting interesting and important work. I look forward to watching Christoffer and his colleagues over the years as they expand on the work we discussed today, and it will be interesting to see how his laboratory develops and grows over time.
Yes, Ken, I definitely agree about Christoffer's work. If you enjoyed this interview as much as Ken and I did, we invite you to visit the STEM Talk webpage where you can find the show notes for this and other episodes at stemtalk.us. This is Dawn Cornega signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others. So please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.